Well, hello again, and welcome to the Science of Childhood Cancer lecture series, hosted by St. Jude Comprehensive Cancer Center. My name is Greg Armstrong, and I'm co-leader of the Cancer Control and Survivorship Program. On behalf of myself and my co-program leader, Melissa Hudson, we welcome you to the 13th week of this 15-week series. The Science of Childhood Cancer is a weekly virtual lecture series designed to engage the scientific community on key questions in pediatric cancer research. Lectures cover a range of topics across the spectrum of basic, translational, and clinical outcomes, including population science and cancer survivorship. We are delighted at the enthusiastic reception of this series, which first began as a 15-week fall session in August of 2020, and has had engagement from thousands of people across the globe. It is my pleasure today to welcome Dr. Jiaming Wang from our own Cancer Control and Survivorship Program here at St. Jude Children's Research Hospital. Dr. Wang is an associate member of the Department of Epidemiology and Cancer Control at St. Jude Children's Research Hospital, with a secondary appointment in the Department of Computational Biology. He has a diverse educational background with an undergraduate degree in biology, a master's degree in mathematics, and a PhD in bioinformatics from George Mason University. Jiaming worked in Silicon Valley as a software engineer before joining the Division of Cancer Genetics and Epidemiology at the National Cancer Institute, where he established himself as an expert in germline cancer genetics. One of his most impactful contributions to the field of cancer epidemiology and genetics was designing the OncoArray for the Genetic Associations and Mechanisms in Oncology Consortium, which he led to a series of major publications around 2017. Jiaming joined St. Jude in 2015 to lead both the operational and scientific aspects of the St. Jude Lifetime Cohort Genomics Project. In 2018, he led the groundbreaking publication on genetic susceptibility to second cancers published in the Journal of Clinical Oncology. Since 2007, he has been an author on over 180 publications, including multiple publications in high-impact journals such as Nature Genetics, Journal of Clinical Oncology, JAMA Oncology, and JNCI. Please join me in welcoming Dr. Jiaming Wang.
Thanks, Greg, for the nice introduction. And the title of my talk today is Genetic and Epigenetic Approaches Towards Understanding Risk of Chronic Health Conditions Among Survivors of Childhood Cancer. I will start with a couple of slides to orient you to survivorship research. Due to advancement of cancer therapy over the last several decades, greater than 85% children diagnosed with cancer will survive 5 or more years. About 13,000 out of 15,000 newly diagnosed cases will become survivors each year. The total number of survivors was estimated to be 500,000 in 2020, which translates to 1 in 750 in the US is a childhood cancer survivor. Survivors are at risk for a broad spectrum of adverse outcomes due to cancer and treatment. Although there are some well-established links between the treatment and the later effects, not all risks can be accounted for by treatment exposures. And that is where genetics and epigenetic factors might come into play. My research primarily used resources from 2 established cohorts of childhood cancer survivors, St. Jude Life and CCSS. Both are hospital-based retrospective cohorts with prospective follow-up. St. Jude Life is a single center enrolling only St. Jude cancer patients, whereas CCSS included 31 institutions. Both included 5 or more year survivors and included patients diagnosed with cancer during the last several decades. Age at diagnosis is younger than 25 St. Jude Life and younger than 21 for CCSS. St. Jude Life considered all diagnoses, whereas CCS included most major diagnosis groups. All treatment information are available for both cohorts. St. Jude Life has both clinical visits and surveys as methods of contact. CCSS is based on survey only. St. Jude Life uses clinical assessment for outcome ascertainment, whereas CCSS use self-reports. However, self-reported second cancer are confirmed with pathology reports. Survivors of germline DNA is blood in St. Jude Life and either saliva or blood for CCSS. The data that I will present today either have already been available or will be made available through our survivorship portal with the URL shown at the bottom of the slide. I will start with likely pathogenic variants in cancer predisposition genes, and then examine polygenic determinants, and finally, likely pathogenic variants in DNA repair genes. Second, for aging biomarkers research, I will cover leukocyte telomere length, epigenetic age, and a GWAS study using epigenetic age acceleration as a molecular phenotype. Third, for epigenetic research, I will show 3 projects, including treatment exposures, social determinants of health, and blood lipids. Let's start with the first published study. To investigate how genetics may play a role in modifying genetics treatment and age-related disease risks, St. Jude Life Genomics Project was started in 2015. The project was a collaborative initiative primarily between Department of Epidemiology and Cancer Control and Department of Computational Biology. Blood-derived DNA was sequenced with whole genome sequencing done at Hudson Alpha and whole exome sequencing at Computational Biology Genomics Lab, directed by John Easton. The study population included 5-year survivors of childhood cancer treated at St. Jude Children's Research Hospital between 1962 and 2010. Frequency-matched community controls were included for comparison. 3,006 survivors were included in the primary analysis. They are largely white, non-Hispanic, with a mean age of 36 years and a mean follow-up of 28 years. St. Jude has established an institutional list of 156 genes associated with cancer predisposition, which included 60 genes that are inherited in an autosomal dominant manner with moderate to high penetrance. These 60 genes were augmented by an additional 96 genes considered to be associated with cancer predisposition. Sequencing data were put through an automated pipeline with over 4,000 candidate variants, triaged by an auto-classifier, resulting in 798 variants. These variants were reviewed by analysts and further reviewed by a panel who provided a final rating of likely pathogenic, variant of unknown significance, likely benign, and benign. All likely pathogenic variants were taken into MiSeq for validation before statistical analysis for variant status and risk of subsequent neoplasms. Overall, we identified 365 likely pathogenic variants in 84 genes. Shown here are 26 most frequent genes with those variants. The prevalence is 6% to 12%, depending on which gene set you look at. 3% of germline variants were found to be mosaic and could be of somatic origin. Based on validation with deep sequencing, we confirmed 4 out of 42, or 9.5%, pathogenic and likely pathogenic variants in RB1 gene are mosaic, and 2 out of 10, or 20%, likely pathogenic variants in TP53 are mosaic. One survivor of soft tissue sarcoma with a mosaic BRCA1 variant with variant allele fraction about 25%, who didn't receive RT treatment but developed subsequent breast cancer. Another survivor of retinoblastoma with a mosaic RB1 variant with VAF around 20%, who developed subsequent sarcoma, which is out of the RT field. The independent genetic contributions to cancer risk were assessed for each subsequent neoplasm and stratifying by irradiation. Among irradiated survivors, rates of subsequent breast cancer and sarcoma were significantly increased for variant carriers. Among non-irradiated survivors, risk of developing any subsequent neoplasms, breast cancer, non-melanoma skin cancer, and 2 or more histologically distinct subsequent neoplasms was significantly increased for variant carriers. Shown here are the genes where a likely pathogenic variant carrier developed subsequent neoplasms. Subsequent breast cancer occurred in 4 genes, subsequent sarcomas occurred in 3 genes, subsequent thyroid cancer in 5 genes, and so on. Survivors with the same subsequent neoplasm carry pathogenic, likely pathogenic variant in different genes, and the same gene confers risk for different subsequent neoplasms. The genetic heterogeneity and pleiotropy pose great challenges in studying germline cancer genetics. Our findings show 12% of survivors carry a pathogenic, likely pathogenic variants in one of 156 genes. If restricted to 60 genes, 6% are variant carriers. It is notable that substantial heterogeneity was observed in subsequent neoplasm within carriers of variants in cancer predisposition genes. Clinically, the high prevalence of pathogenic, likely pathogenic variants supports referral of all survivors for genetic counseling. And clinical genetic testing should be prioritized for high-risk groups. Move on to the contribution of polygenic determinants to risk of subsequent breast cancer and subsequent thyroid cancer. The genetic risk of breast cancer in the general population has been extensively studied. The full picture of its allelic architecture includes rare, highly penetrant variants, uncommon, moderately penetrant variants, and common, low penetrant variants. Although individually these common variants contribute very little to risk, when we construct a polygenic risk score based on some weighted sum of the risk alleles carried by each person, the combined effect could be substantial. The polygenic risk score follows a normal distribution. The quintile cutoffs were very close to the expected numbers based on the minor allele frequency from uncorrelated control population, which suggests the genetic background resembles each other. The overall standardized incidence ratio is 6.7, comparing this high-risk survival cohort with the general population using SEER data, ranging from 3.7 for survivors with PRS in the lowest quintile to 11.4 in the highest quintile. The overall absolute excess risk was 1.7 per 1,000 person-years of follow-up, ranging from 0.8 to 3.1 within each quintile. Here, we observed the rate of cumulative incidence of subsequent breast cancer differed by polygenic risk score. By age of 45, rates of cumulative incidence were 15.5% for survivors with PRS in the 5th quintile, 4.2% for 3 middle quintiles, and 2.8% for the 1st quintile. Statistically significant effects were observed in the joint modeling of monogenic and polygenic associations with subsequent breast cancer risk. When stratified by chest RT, pathogenic, likely pathogenic variant carriers have a 10-fold increased rate versus non-carriers. Survivors with PRS in the highest quintile have a 3-fold increased rate comparing with survivors with the PRS in the lowest quintile, with significant trend across quintiles for irradiated survivors. The association is not statistically significant among chest non-irradiated survivors due to limited sample size. Stratified analysis by age group showed that polygenic risk score confer risk among younger female survivors with age less than 45 years specifically. In summary, rate of subsequent breast cancer was significantly increased for survivors carry a pathogenic, likely pathogenic variant or having a higher polygenic risk score. Based on PRS, we were able to identify additional survivors with risk equivalent to monogenic variants in our study population. Clinically, we anticipate utilizing common low-risk loci in combination with rare high-risk variants and other clinical risk factors may inform a better strategy for subsequent breast cancer risk stratification. We also evaluated the contribution of polygenic determinants for another common subsequent malignancy, thyroid cancer. Using Senju Life as a discovery cohort, we found per standard deviation increase of standardized PRS had an increased rate of subsequent thyroid cancer with a relative rate of 1.57 overall, 1.67 for survivors with NEC-RT, and 1.36 for survivors without NEC-RT, after adjusting for other risk factors listed in this table. Using CCSS as a replication cohort, we found post-standard deviation increase of standardized PRS had an increased rate of subsequent thyroid cancer with a relative rate of 1.51 overall. Risk prediction models were built using Central Life cohort, showing that integration of PRS improved the performance. For example, the AUC was improved from 0.82 to 0.83 at age of 40. In CCSS, the integrated prediction model was validated with the AUC improved from 0.71 to 0.73. Although the integrated prediction model improved with statistical significance, we see the magnitude was only modest. However, we argue that PRS can be used to stratify subsequent thyroid cancer risk for screening prioritization. In general, additional stronger evidence needs to be provided to support the polygenic clinical testing be adopted as a common practice. Move on to the final genetic project, investigating the variants in DNA repair pathways and risk of subsequent neoplasms. We think cancer survivors are an ideal population to study the contribution of DNA damage repair pathways to adverse health outcomes, including second cancers, because DNA in survivors has likely experienced damage earlier than and at much greater magnitude than that expected during normal cellular aging. We expanded our study population to 4,402 survivors in Central Life cohort by sequencing additional 1,400 younger survivors. Shown in this table are demographics and treatment information. We focused on 127 genes from 6 major DNA repair pathways, including homologous recombination, nucleotide excision repair, mismatch repair, base excision repair, non-homologous end joining, and Fanconi anemia pathway. Of 4,402 survivors, 495 or 11.2% developed 1,269 subsequent neoplasms. 538 pathogenic, likely pathogenic variants in 98 DNA repair genes were found in 508 or 11.5% survivors. So this table shown here summarizes the results for pathway-based association analysis, including 36 combinations of pathway and SN outcome, 6 rows and 6 columns. 6 rows are the pathways and 6 columns are the different subsequent neoplasm outcomes. Variants in certain DNA repair pathway are associated with rates of subsequent neoplasm. For example, likely pathogenic variants in homologous recombination genes are associated with increased rates of subsequent breast cancer and sarcoma. Likely pathogenic variants in nucleotide excision repair genes are associated with increased rate of subsequent thyroid cancer. For overall significant associations, we further stratify by treatment exposures. Strata corresponding to the highest dose of RT or chemo agents had higher relative rates. A further look at cumulative incidences with 4 curves in each plot showing the combinations of variant status, yes or no, with treatment dose, high or low. Take a closer look at the one example for the cumulative incidence of breast cancer using a 10H as time scale. The high-flying red curve corresponds to a group of survivors who had likely pathogenic variants in homologous recombination genes and also received chest RT with dose greater than or equal to 20 gray. In summary, we found that female survivors who are pathogenic, likely pathogenic variant carriers in homologous recombination genes and treated with high dose of chest RT or anthracyclines had increased rate of subsequent breast cancer. Survivors who are pathogenic, likely pathogenic variant carriers in homologous recombination genes and had treated with high dose of alkylating agents had increased rate of sarcoma. Lastly, survivors who carry pathogenic, likely pathogenic variants in nucleotide excision repair genes and received high dose of NEC-RT had increased rate of subsequent thyroid cancer. Our study provides novel insights regarding the genetic contribution in combination with the known treatment factors, and the findings really support our initial hypothesis that a combination of DNA repair deficiency with genotoxic exposure increases the risk of subsequent neoplasms. These findings may facilitate identification of high-risk survivors and inform personalized cancer surveillance and prevention strategies. Let's switch gear to the area of aging biomarker research, where I'm going to first talk about leukocyte telomere length. We know hallmarks of aging are multifaceted and include different biological mechanisms. My colleague, Dr. Kiri Ness, pioneered aging research in survivorship, reported frailty as a sign of aging acceleration among survivors. Inspired by this, I've started looking into mechanisms including telomere length, mitochondrial dysfunction, epigenetic alteration, and genomic instability. Telomere is a nuclear protein structure at both ends of a chromosome to protect its integrity. Telomere inevitably becomes shorter during normal aging, and the age-dependent attrition can be influenced by both genetics and non-genetic factors. Childhood cancer survivor could be more vulnerable to telomere attrition than general population because they have experienced biological damage caused by genotoxic cancer treatment like radiation and chemotherapy. As an aging biomarker, telomere is also shown to be associated with age-related diseases such as cardiovascular disease, cancers, and mortality. We derived the leukocyte telomere length using whole genome sequencing data from St. Jude Life. And we found leukocyte telomere length in survivors was significantly shorter overall and across diagnoses compared to controls. When projecting onto the linear regression lines of leukocyte telomere length against age, the leukocyte telomere length of 6 kilobases corresponds to age of 37 years among survivors and age of 48 years among controls, suggesting acceleration of aging by 11 years. However, the age-dependent attrition rate was only modestly greater among survivors. By comparing leukocyte telomere length between survivors with and without certain cancer treatment, we found survivors exposed to chest or abdominal RT, glucocorticoids, or vincristine had shorter leukocyte telomere length than non-exposed survivors. In addition, leukocyte telomere length was significantly shorter among survivors with unfavorable health behaviors among the younger group aged 18 to 35. We found there were negative associations between leukocyte telomere length and 14 common non-neoplastic conditions, and a positive association between leukocyte telomere length and the occurrence of subsequent thyroid cancer. For example, prevalence of obesity, the most common condition, is negatively associated with leukocyte telomere length. The results suggest that leukocyte telomere length may be a potential mechanistic target for future intervention studies designed to prevent or delay onset of chronic health conditions in childhood cancer survivors. Now let's move on to another aging biomarker, epigenetic age. There are at least a dozen epigenetic clocks have already been proposed. Here listed are 4 popular ones. Hannum and Horvath are the first generation of the clocks selecting CpGs to predict chronological age with smaller number of predictor CpGs. Levin and Grimage were among the second generation attempting to model physiological measurements and health outcomes with larger number of CpGs. We adopted Levin's clock, phenoage, to calculate the epigenetic age in our first published study. The concept of epigenetic age acceleration can be illustrated with this cartoon. The solid blue line shows how epigenetic age changes with age. The rate is very fast in the very first year of life. Afterwards, it decreases gradually in a nonlinear manner until about 20 years of age, and then gets to a slower constant rate. Individuals above the line had positive epigenetic age acceleration and are older than their peers at the same chronological age. Conversely, individuals below the line had negative epigenetic age acceleration and are younger than their peers at the same chronological age. The correlation between epigenetic age and chronological age is high, with Pearson correlation coefficient of 0.88 for both survivors and controls. However, the age slope, that is the annual change of methylation age, is 1.24 versus 1.08, which is significantly different. We found epigenetic age acceleration is much higher in survivors than controls. Survivors of Hodgkin lymphoma seems to have the highest epigenetic age acceleration across all primary diagnoses. Survivors with chest RT, abdominal RT, pelvic RT, or exposures to 3 different chemo agents had significantly elevated epigenetic age acceleration than non-exposed survivors. We compared survivors with 5 different modifiable health behaviors, including tobacco use, diet, physical activity, alcohol use, and resistance training. Due to the multifactorial nature here, we defined 3 groups, including favorable, 4 or 5 good, intermediate, 2 or 3 good, and unfavorable, 0 or 1 good, based on these 5 components, and observed that epigenetic age acceleration trended up among survivors with favorable, intermediate, and unfavorable health behaviors. 6 chronic health conditions were significantly associated with epigenetic age acceleration after adjusting for other factors. There are hypertension, myocardial infarction, obesity, peripheral sensory neuropathy, obstructive pulmonary deficit, and obstructive sleep apnea. Finally, let's look at 2 aging acceleration models. And we can see the aging trajectory for survivors varies depending on the dynamics of molecular marker. Telomere length showed a pattern of accentuated aging with the initial hit, but a parallel age slope. And epigenetic age acceleration, epigenetic age showed a pattern of accelerated aging with a faster pace or steeper age slope. To conclude, epigenetic age acceleration is higher among survivors than controls. Epigenetic age acceleration is elevated among survivors exposed to certain treatment or unfavorable health behaviors. Epigenetic age acceleration is positively associated with increased rates of certain chronic health conditions. Clinically, epigenetic age acceleration can potentially be targeted for future intervention studies. Move on to genetic association study of aging biomarker. 2 determinants underlying the biological aging. Here, we discovered a strong HLA locus associated with epigenetic age acceleration based on Hannum clock after adjusting leukocyte cell subtypes. Interestingly, the replication was strong in an additional set of survivors, but not in controls. The linkage disequilibrium block for the top hits in this region suggested that sequential conditional analysis may be needed to tease out potential independent signals embedded in this strong association peak. We also found an association at CELP gene locus using epigenetic age acceleration based on Horvath's clock. As you may know, CELP gene is not only a marker for aged hematopoietic stem cells, but also is functionally involved in age-associated HSC functional decline. The initial discovery was borderline genome-wide significant, but it got nicely replicated in both survivors and controls. The LD block looks much tighter compared to the HLA locus, suggesting a single causal variant in this locus. In a recently submitted grant proposal, we plan to study multiple aging biomarkers, including integrating the driver mutations of clonal hematopoiesis of indeterminate potentials that Dr. Jingwei Zhang and her postdoc are currently characterizing and validating. Let's move on to epigenetic research with the first published study looking at DNA methylation and treatment exposures. There are two columns in this diagram. If you are a biostatistician or data analyst, please look at the right column. Otherwise, you only need to look at the left column with me. First, we performed treatment-specific epigenome-wide association study to identify treatment-associated CpGs, and then search for the subset of treatment-associated CpG sites also associated with cardiometabolic conditions, followed by multiple mediation analysis. In the end, we perform a single mediation analysis using combined methylation score for the CpGs that are individually significant mediators. By performing multiple treatment-specific EUAs, we identified about 1,000 CpG sites associated with one or more cancer treatments at epigenome-wide significance level. Among 652 CpG sites associated with chemotherapy exposure, there were 198 CpG sites associated with two or more chemotherapy agents, and the remaining was specifically associated with alkylating agents, anti-metabolites, asparaginase enzymes, epiphthalatoxins, and corticosteroids. Among 462 CpG sites associated with RT exposure, there were 273 CpG sites associated with two or more region-specific RT treatment, and the remaining are specifically associated with brain RT, chest RT, abdominal RT, and pelvic RT. DNA methylation mediates the association between treatment exposures and cardiometabolic conditions. As an example, using a summation of the methylation levels of 4 CpGs as a single mediator variable, 70% of mediation was achieved for the effect of abdominal RT on risk of dyslipidemia. In summary, a couple of points to make. Cancer treatment exposure exerts persistent impacts on DNA methylation variations, and treatment-associated DNA methylation sites may mediate the causal pathway from specific treatment exposure to certain cardiometabolic conditions. Beyond early treatment exposure, survivors, like everyone else, will experience lifelong exposure to socioeconomic positions as well as associated health behaviors. Social epigenomics is an emerging multidisciplinary research field gaining some attractions in the general population. Based on this rationale, using treatment EUAs as a proof of feasibility and also to show rigor of our analytical capability, my colleague, Dr. Ai-Qian Huang, and myself successfully obtained a V Foundation grant to conduct a social epigenomic study in childhood cancer survivors with these three specific aims. First, characterize health disparity using measures of social determinants of health. Second, perform epigenome-wide association analysis of SDOH and further evaluate the functional implication of SDOH-associated CpGs. Third, evaluate the mediation of DNA methylation for the effect of SDOH on CHCs. We proposed a social epigenomic framework and hypothesized that social determinants of health and associated health behaviors elicit modifications in DNA methylation that lead to altered gene expression and ultimately result in the development of CHCs. The analytic approach is straightforward. First, do an SDOH exposure EUAs, and then do an outcome UAS. In the end, take the intersection of significant CPGs for further mediation analysis. Now let's first look at the SDOH exposure UAS. This circus plot shows UAS results of educational attainment, red outside circle, personal income, green middle circle, and area deprivation index, blue inner circle. As expected, quite a few association peaks were overlapping across these three UAS. For example, the common association peak at chromosome 3 harbors several genes well known as smoking-related genes, such as CPOX, GPR15, and CLDND1. 13 CPGs were associated with all three SDOH factors and resided at pre-tropy loci featuring cigarette smoking-related genes. And their effects got attenuated when smoking was adjusted in the model. Stratified analysis in non-smoker would help disentangle the effects from different exposures, which may be suggested by the manuscript reviewer we will see. To infer causality beyond association, we conducted the bidirectional Mendelian randomization analysis. First, we used the PRS generated from 74 education attainment-associated SNPs and found only one CPG that was statistically significant, suggesting many of our UAS findings may not be the consequence of SDOH factors. On the other hand, to evaluate potential causal effect of CPG hits on SDOH, we found that PIs derived from 14 independent methylation QTL was associated with educational attainment. This suggests a reversal causal impact and a possible feedback loop between educational attainment-related exposures and DNA methylation level of this particular CPG site. To conclude, DNA methylation signatures, many but not all, resembling the effect of tobacco use, are associated with SDOH factors among survivors of childhood cancer, suggesting that biologically distal SDOH factors may influence the epigenome and subsequently survivors' health. Future longitudinal study with repeated measurements of DNA methylation may be able to help with the causal inference. Lastly, let's look at the UAS of blood lipids. Blood lipids are well-known risk factors for cardiovascular disease. On one hand, DNA methylation could change in response to blood lipids level as an internal exposure. On the other hand, DNA methylation may also have an effect on lipids levels by regulating expression level of genes in lipids metabolism. We took the time point when blood was drawn for DNA methylation assay to split the longitudinal clinical data into an exposure UAS and an outcome UAS between DNA methylation and lipids traits. When lipids were modeled as exposure, epigenome-wide significant hits included 84 for high-density lipoprotein cholesterol, 55 for triglycerides, and 3 for total cholesterol. Most of the hits are lipids type specific. When lipids were modeled as outcome, epigenome-wide significant hits included 5 for HDL, 4 for LDL, 98 for triglycerides, and 4 for total cholesterol. Similarly, most of the hits are lipids type specific. In summary, based on UAS catalog, most of our statistical significant findings seem to be novel. Almost all of the findings are null in survivors of African ancestry, which suggests epigenetic associations are specific to survivors of European ancestry. Further validation using cardiac study is currently on the way to find out if any of these associations are survivor specific. Additional ongoing analysis include correlations between DNA methylation and gene expression, as well as Mendelian randomization analysis using methylation QTL or lipids-associated genetic variants as instrument variable for causal inference. My lab aims to continue this line of research with a focus on identifying novel and informative clinical biomarkers using multi-omics data, including genetics, understanding of the pathophysiological trajectory, and using this knowledge to design pharmaceutical or behavior coaching to prevent or delay the onset of chronic health condition in our vulnerable cancer survivor population. I will end with acknowledging many people in our collaborative research, particularly program leaders, my collaborators, data analysts, and as well as my trainees. And finally, the funding sources, as well as our study participants. I really miss the time when we have face-to-face discussions. These are old pictures taken in 2018 and 2019, and nothing in 2020 last year. So at that time, we were so close to each other without wearing masks. Thank you, and I'm happy to take questions you may have.
Well, Jiaming, thank you. That was a fantastic lecture, and thank you for sharing your most recent data with us. I'd like to remind all participants to submit questions through the chat box, specifically submitting it to the all panelists group. All right, Jiaming, if you're willing and ready, we can dive into a few questions. Let's start with this. A key component of studying epigenetics among survivors is having longitudinal data. What would be your preferred interval for collecting samples for performing methylation studies within this high-risk population? So if you had the opportunity to do longitudinal evaluation, when would be the next time you'd want to see them?
Well, that's a really great question. So actually, in the grant that I submitted, I propose a 5-year interval, and which also matches with sort of the time point we have for essential life visits, clinical visits. So we do have samples biobanked at these different time points. And I expect that 5-year should be enough to accumulate sort of the effect from any type of exposures.
Okay, great. All right, so did you sequence any general population controls? And if you did, what was the prevalence of the pathogenic or likely pathogenic variants for cancer predisposition in that population as compared to the group you showed us?
Yes, that's a great question. So I couldn't have enough of time to sort of squeeze in that slide, but we did sequence 340 in the first batch, the community controls for Central Life cohort, and we sequenced additional 106 in the second round of whole genome sequencing. So we have about 450 controls. And the prevalence in the controls, we observed it's about 0.5 to 0.6%. So it's about, if you compare survivors with the controls, so it's about 10-fold increase. So it's a 6% for the 60 genes in the survivor cohort and 0.6% in the controls. And also I want to elaborate a little bit. And actually the Dr. Jingwei Zhang led the publication New England Journal of Medicine in 2015 and published, it's about reported 8.5% for the 60 genes for the diagnostic case series. And we reported 6% using the exact same gene panel for the survivor cohort, which probably implying that the genetic predisposition could be related to early mortality. In fact, Dr. Jun Yan in Pharmaceutical Science published a paper in JCO reported indeed the TP53 is implicated in the prognosis of the cancer patients.
Great. Thank you, Jiaming. Let's talk about clonal hematopoiesis. There's a question there. It's an area of high interest. And what do you see as the risk for CHIP among these survivors?
So that's a very good question. So in the general population, there are a lot of study conducted already. So the initial reason we have held whole exome sequencing designed sort of paired with whole genome sequencing is to give enough of sequencing coverage so that we can have a first pick into the clonal hematopoiesis. That's Dr. Jingwei Zhang first designed this study. Later on, during the work for the first paper, when we report the genetic susceptibility for second cancer, we indeed look into that, as I showed in a couple of slides, showing that the mosaicism in the cancer predisposition gene. But nowadays, she and her postdoc already started looking into using the deep sequencing strategy, really looking into the driver mutations. And based on our preliminary data, I already tested, showing, validating, whatever already people are seeing in the general population, like looking at we observed a strong association between the clonal hematopoiesis chip mutation carriers have a much higher increased risk for myocardial infarction. So that's we're going to finalize currently is validating the chip. All the chip cores use orthogonal experimental methods, and once we get that finalized, we certainly will expand and look into other chronic health conditions.
Well, then following on that, we've talked about epigenetic age, telomere length, and now chip. There's still other mechanisms, underlying biologic mechanisms of aging. Do you propose to investigate any others, or what do you think are the next step for evaluation?
That's a great question. So the answer is absolutely yes. So not only expand the aging biomarker by including, beyond the epigenetic age acceleration, the telomere length, and looking into chips and also mitochondrial copy number and heteroplasmy, and look at the chromosomal abnormality. For all of these, not only expand that portfolio, also sort of dig deeper into each of them. For example, I'm interested in looking into at the single cell level, look at epigenetic age acceleration, sort the PBMC and look into whether there is a cellular heterogeneity in terms of how fast at the cell level, at single cell level, how fast the epigenetic age look like.
Sounds great. All right, here's a question from Lisa Diller. Is mosaicism for the pathogenic variants or the likely pathogenic variants in cancer predisposition associated with an intermediate phenotype in terms of primary disease outcome and second cancer risk?
The germline mosaicism, yes, we looked at the second cancer risk. It's included. Because of the number, we cannot really sort of look at the subset. We just look at it together, and a couple of examples sort of as at the descriptive level, I showed that germline mosaicism, the mutation carriers indeed developed second cancer. And I emphasized that both of them actually do not have a radiation treatment. So most likely caused by the genetic reasons.
So here's a question from Susan Weiner. She says these data are exciting in regards to prevention strategies for late effects of treatment. It appears that the work so far does not include patients with primary or secondary brain tumors. Perhaps you could address that. And if that's true, can you comment on your work as it might apply now or in the future for these brain tumor patients?
So I think we do include survivors of brain tumor, the CNS in the Central Life cohort. So remember, Central Life cohort consider all diagnoses. Although there's maybe the cure rate is low. People may die from the diagnosis, unfortunately, and we couldn't include those people. But in our study, we have the CNS survivors.
Great. All right. The integration of multiple aging biomarkers seems fascinating. You've commented on one or two independently, but how do you imagine integrating these? Are they correlated? Can you put these into models together? I'd be interested to see how you put all this together.
Sure, yeah. So we individually sort of focused, and we generate new data, publish results, and build our knowledge, sort of understanding in the survivors, the well-known, for example, the leukocyte telomere length. And one of the person who reviewed the paper actually wrote a research highlight on our paper, emphasizing this is really exciting findings and looking into the survivors. So, and we published epigenetic age acceleration. And recently, Dr. Kiri Ness also published a paper in the JNCI. It is in press, looking at mitochondrial copy numbers. So yes, we looked at the correlation between these as expected. So they are weakly correlated because the aging mechanism is really multifaceted, and they inform each other, but not one can replace the other. For example, just give you an example. When I looked at a model sort of using the LTL as the outcome and plugging the age, plugging other factors, and also adding the epigenetic age acceleration. And we found the epigenetic age acceleration after adjusting for other factors. It is associated, it's strongly associated, statistical significant associated with leukocyte telomere length. And in fact, when I look deeper into the correlation, so we all know that the chronological age is the strongest single predictor of leukocyte telomere length, and which only explains about I think 3% of total variation. So telomere is very dynamic, and it can change all the time. So, when we also look at epigenetic age acceleration, it explained additional 2%. So if you rank sort of the predictors of leukocyte telomere length, if age ranks the first, in my model, epigenetic age comes the second. So definitely, when we put together, we see some synergistic and also redundancy, and we just need to be careful when we integrate all of these to inform the risk of chronic health conditions.
Yep, that's good. So here's an important question. Are you thinking of ways to intervene in order to slow down aging? This is certainly provocative work, but certainly you're also thinking, can we intervene on this? Can we stop it? I'd love to hear your thoughts on that.
Yeah, I mean, first of all, I'm not a clinician. I couldn't say that, but as I just showed on my last slide, I'm realizing the promise of precision prevention medicine. So that's, a lot of people have shared the same goal with me. So I think, our research is really, we conduct clinical research and we publish the results and sort of build up the knowledge base. But it's really up to the clinician and the practitioners sort of to gauge on that whether these are ready for prime time clinically. So certainly, we are working towards that goal. And for example, epigenetic age acceleration, epigenetics is reversible. So industry, pharmaceutical industry, and people are developing drugs targeted at methylation modification. Hopefully, we can rejuvenate ourselves and making ourselves much younger than the chronological age, and sort of reduce the burden of chronic health conditions. Yes.
Yes. We hope to find the fountain of youth, I guess, hopefully in our lifetime. Yeah. So just a couple more questions, Xiaoming. This is in regard to the confounding effect of the primary cancer diagnosis. So regarding later outcomes in survivors, and we know they're dependent on the treatment exposures, do you feel like you've really eliminated the influence of the primary cancer and cancer type in these analyses? Or is it still a confounding?
Well, yes or no. I think we couldn't adjust because it's too many, just from a statistical point of view. But we adjust for treatments, and treatment to some extent is a surrogate for primary diagnosis because they usually follow the same protocol. So I think, for example, just, Hodgkin lymphoma, we see the highest epigenetic age acceleration in Hodgkin lymphoma. And why? And when we ask the question and we link to the treatment effect, we found that ChestRT is the most strong, I call it potent exposure. 90% of Hodgkin lymphoma patients will receive ChestRT. So that sort of linked them together. I think eventually we really need larger sample size or focus on a specific primary diagnosis to study, sort of to tease out the effect from the cancer diagnosis.
Great. Well, Xiaoming, it's the top of the hour, so I want to thank you for your outstanding talk, and we need to officially close the event. But I want to invite all who are interested to stay on because you're going to be with us for another few minutes to answer some additional questions. I'd also like to mention that you should mark your calendars for next week, where Dr. Kim Stegemeyer from the Dana-Farber Cancer Institute will be speaking. Her talk is titled A First Generation Pediatric Cancer Dependency Map. So thank you for your attendance, and for those who'd like to stay, we'll ask a few additional questions. Xiaoming, here's one. In childhood cancer survivor populations, do you think whole genome sequencing is the standard for genetic research, or can SNP array data still be recommended?
I think we are moving towards sequencing technology, everybody in the field. When I decided to join St. Jude in 2015, one of the things that attracts me, as Jingwei called me and on the phone and talked about this data set, and we are going to building this up. And I really get excited. I think this is a technology drive this, and also, I had a lot of years of working on the sleep array. I designed the OncoArray, which I feel really proud of. But the array is fixed content. That's a limitation we cannot conquer, and it's the content was based on the sort of cataloged known variants. Whole genome sequencing certainly triumph over. You can get the rare and common. And also the good thing is the pricing is dropping, so the cost won't be a big deal if we choose this technology. So I would vote for WGS.
Sounds great. There's a quick question about 5-year survivors, and both studies required 5-year survivorship. If you had been able to include a population that were prior to 5 years, or 0 to 5 years, do you think they would have changed results in any way?
Oh, that's a great question. So I think we have the HEC clinics here at St. Jude. If we have the samples, I believe we have the samples, but just a matter of if we initiate anything and to do this research, it will be great to compare sort of the patient still in the, within sort of after treatment, but before the 5-year survival sort of enroll in the St. Jude Life. We can certainly look at this group of patients.
Okay. Here's another one. Have you correlated self-reported health-related quality of life with biologic markers of aging? There's a note here that a prior look at self-reported health utilities from CCSS showed similar shifts, similar to your data, where younger survivors looked older than they should based on their health-related quality of life. So curious if you've looked at that as an outcome.
I haven't yet, but this is a really great suggestion. So I can look at it. I think the self-reports, what I, based on my conversation with Ai-Chen Wan, my colleague here, we discussed about the quality of the data self-reports, and we discussst about for example the smoking, self-reported smoking. It's underreported, and in fact. We can even use blood-based methylation data to sort of objectively say that you are smokers and you are non-smokers, rather than rely on your questionnaire results. So, yeah.
Okay. All right, there's one last question. You ready?
Yes.
Okay, here we go. The epigenetic clock derived from the general population seems to work in childhood cancer survivors. I think the question here is, do we think we need an epigenetic clock for survivors, specifically built for survivors from DNA methylation data?
Oh, yeah. So, we, when we study survivors, I mean, really, we sort of look at what people have already done in the general population, right? So we borrow concepts and whatever the findings and see into our data. And that's what we learned sort of epigenetics initially when I developed grants with Dr. Kiwi Ness, and I proposed the epigenetic clock in the beginning. And we had money, sort of seed money to do only 300, right? So we used, at that time, Horvath's clock and calculated show as a preliminary data. But, I always had that in my mind. And we knew that childhood cancer survivors, they experienced the treatment effects, and this is really strong. And during the crucial sort of developmental stage, and they should have impact on the methylome. And so Zhenghong Li, who is doing his PhD with me for the dissertation, he's a lead analyst in our department. And one of his projects is to use machine learning. We're going to use ElasticNet, Lasso, and to select the predictors. Indeed, he already made rapid progress, and we selected 700 CPGs, and we found, higher correlation, which is not surprising. We we got 0.99% as a correlation compared to 0.88. That's what I should use others clock. And recently, I also suggest him to look into deep learning method using neural network based to derive, I call it survivor-specific epigenetic edge clock. Yes.
Great.
It will be very exciting.
Oh, that's good. We look forward to that. Well, Xiaoming, thank you so much for taking the time to share your work with us. We've spent decades now looking at the epidemiologic associations between treatments and outcomes, and your work really goes a long way to help us understand the underlying mechanisms, but also create some exciting targets for the future for prevention and intervention. So thank you so much. And thank you everyone for attending our lecture today. Have a great day.
Thank you, Greg. Thanks, everyone.
Take care.